So, hello everyone. Welcome today to our video. Today we'll be discussing the histological organization of the lymphatic tissue. Remember the last video we did was on the histological organization of the cardiovascular system. So, once again, karibu. So, what is the lymphatic tissue? What is this lymphatic tissue that I talk about? So, the lymphatic tissue is an aggregation or tissues or organs which are mainly consist of dense accumulation of lymphocytes. So we have tissues, organs, which consist of dense accumulation of lymphocytes. That's the reason from the word lymphatic, we have lymphatic fluid, lymphocytes in that regard, lymphatic tissue. And these lymphatic tissues, they contain reticular fibers and reticular cells, which form the capsule or the stromal component in which now the lymphocytes lie inside them. And these lymphatic tissues may be described as diffuse lymphoid tissues, might also be described as dense lymphoid tissue. So in terms of this dense lymphoid tissue, that is something like mucosal associated lymphatic tissue, usually abbreviated as mucosal associated lymphatic tissues, where we have solitary nodule, just one nodule alone, it's known as solitary nodule, one lymphatic nodule, one solitary lymphatic nodule. Other ones can be now aggregations of these nodules, aggregations of the lymphatic nodules, some of them the Peyer's patches, located in the terminal part of the ileum. Other parts, they will be located in the GI, that is the gastrointestinal system, the urinary system, and even in the respiratory system. Others will be located in the vermiform appendix, so that is the lymphoid tissue in the vermiform appendix. Others in the Walde's ring. Remember the components of the Walde's ring? We had the adenoids at the roof of the pharynx. We had the tubal tonsils. We had the palatine tonsils, there are two. And then we had the lingual tonsil, giving us a Walde's ring. So it's just a ring comprising of lymphatic structures. And then you have now discrete or solid lymphoid organs, talking about something like the thymus, the spleen, which is the largest lymphoid organ, and the lymph node. These lymphatic organs may be classified into two. We can have primary lymphoid organs or central lymphoid organs. So primary, also known as central lymphoid organs. And also we can have peripheral, also known as secondary lymphoid organs or lymphoid tissues. So why are they called central? Why are they called primary lymphoid organs? Because they are site of immune cell production. And which cells specifically? T cells and B cells. So thymus, T cells, bone marrow, B cells. What about the peripheral immune organs or secondary lymphoid organs? These are the organs or the lymphatic tissues where immune cell function. So after you have the B cells and the B and the T cells being produced from the thymus and the bone marrow, they come into now these peripheral immune organs. Talk about now the spleen, the lymph node, the mucosal associated lymphatic tissues, the Walde's ring, the palatine tonsil, the adenoids, the Peyer's patches. That's why you have all this immune cell functioning. So you'll divide the immune system into two. Remember, we usually have innate immune system and adaptive immune system. Under adaptive immune system is when we'll find these lymphocytes. And in the lymphocytes, we have B and T lymphocytes. To be specific now, B lymphocytes will be concerned with humoral immunity, while T lymphocytes will be concerned with cell-mediated immunity. We talked about the components of the lymphoid organs, talked about the mucosal associated lymphatic tissues. We talked about the lymph nodes. We talked about the spleen. So remember, for the lymph nodes, they are located in various places. So we can have supraclavicular lymph nodes, inguinal lymph nodes, axillary lymph nodes. You can have popliteal lymph nodes. There's so many lymph nodes, the body, mediastinal, celiac, mesenteric lymph nodes. There's so many types of lymph nodes, and actually they are classified according to regions. We can have spleen, the largest lymphoid organ. We can have the thymus. We can have the palatine tonsil as one of the components of the Waldeus ring, but also all the components of the Waldeus ring will form its components here. For the thymus, let's now start uncovering the specific organs. So this thymus is a specialized primary lymphoid organ. That's a keynote point. Keynote point out is that it's a primary lymphoid organ that is located in the anterior mediastinum or superior mediastinum. Then this thymus, as you grow, it degenerates. It becomes replaced by a fibrofatty tissue. So at around puberty, at around the age of 6, as you grow old, due to the adaptive immune system coming in, the function of the thymus decreases and becomes replaced by the fibrofatty tissue. Functions of the thymus is that it enables the development of the immunocompetent T lymphocytes. That's one function. The other function is that it leads to much supply of T cells into circulation. The other function is that it leads to development of the immunoregulatory self-tolerance by negative selection and positive selection that happens in the thymus. We also have secretion of hormones and cytokines. So by the function of having some cells, the plasma cells, the lymphocytes will produce cytokines. Some of them will be inflammatory, some of them will be pro-inflammatory cytokines. In terms of hormones, it will produce thymosin, thymulin, and thymopoietin hormones, which we'll see in a few minutes. When we're talking about now the components of the blood-thymus barrier, remember you have a barrier in the thymus known as the blood-thymus barrier. And this blood-thymus barrier will comprise of the capillaries. At this point, remember the capillaries should be continuous capillaries, perivascular connective tissues, and epithelial reticular cell layers. So for these capillaries, some of them will be continuous, some of them will be fenestrated, and some of them will be sinusoidal. Remember, the function of the thymus at this point is to allow flow of blood in it. And that's the reason why most of the capillaries in thymus, they will be sinusoidal because this is the reticular endothelial system. So most of the capillaries in the thymus will be sinusoidal types of capillaries. So by the layers now you'll have the layer will have capillary, perivascular connective tissue, or the connective tissue between the capillaries and these epithelial reticular cell layers. So you can have 3 layers. You can also classify it into 5 layers. Here you have the endothelium of the capillary. You can have the basement membrane of the capillary. You can have connective tissue between the two. Then you can have the basement membrane of the epithelial reticular cell, and then finalize now with the epithelial reticular cell layer. So those are 5 layers of the blood thymus barrier. What the function of blood thymus barrier? Just preventing first of all is filtration. Remember, filtration also allowing the T cells to get into circulation and also to do the function of filtration. What about the histological organization of the thymus? You'll divide it into two. That's an image showing us the parenchymal aspect of the thymus. But before then, I'd like us to take us to the stroma of the thymus. So this stroma of the thymus, you'll divide it into two. So remember, this stroma is a connective tissue that is, first of all, covering the whole of the thymus and also sending in trabeculi, sending in septi to divide this thymus into various parts. One part of it is that you have the capsule, which will now cover the whole thymus. So it's a thin capsule and completely covers the whole frame of the whole thymus. Then this capsule will send in interlobular septi or trabeculi to divide the lobes into lobules, to divide completely the thymus into lobules. And this capsule, this septi, this trabeculi, they will carry blood vessels, they will carry nerves. In addition to that, this stroma, they won't be just elastic fibers, they'll be epithelial reticular cells. Can you take note of that? Only the thymus will have the epithelial reticular cells forming the reticular formation, forming the cups, forming the complete interlobular septi of the thymus. So doing that, it will divide the parenchyma into lobes and lobules. What about the parenchymal aspect? So for the parenchymal aspect, it has a cortex, a darker staining component. So this is it, the darker staining component and a lightly staining part. So the lightly staining part is now the inner medulla. So the outer darker staining part and an inner medulla which is slightly staining. So the inner medulla has Hassel's corpuscle. So this is a Hassel's corpuscle, lighter staining, and then it has an outer part, the cortex, which is slightly staining. So cell types in the cortex will have mature T cells, reticular cells, and macrophages. So three cell types in the parenchyma. What about the medulla? For the medulla, it will comprise of now the Hassel's corpuscle, which is a characteristic of the thymus. Also, it will comprise of hormone secretions, which is now the thymosin, the hemopoietin, and thymulin. So kindly take note of that is a histological organization of the thymus. In terms of the slide, I can be able to summarize the whole of it using this slide. This is a thymus, which is a central lymphoid organ because that's where the maturation and the development of the T lymphocytes happens. You have a capsule. So this is a capsule. For the capsule, it will be thin at this point, connective tissue. It will cover the whole of the thymus. To do sending trabecula to cover the thymus, and these trabecula will be the epithelial reticular cells, which form the blood thymus barrier. And these epithelial reticular cells will be connected to one another using the desmosomes, as you can see here. So these are desmosomes connected to one another for communication. So these epithelial reticular cells on the trabeculae, they will divide the thymus into lobules completely. For example, you can see this. And that takes us, actually, that takes us to this lobule, which will be separated completely. So you have an outer staining part, which is an outer staining part of the thymus, which is the cortex, and an inner staining, lightly staining part, which is the medulla with Hassel's corpuscle. So you can see this the Hassel's corpuscle here, another Hassel's corpuscle here, another Hassel's corpuscle here, another Hassel's corpuscle there. So kindly take note of this. We said blood, thymus barrier, which will be made of them. Now the reticular cells, the capillaries to be specific, the sinusoidal capillaries, and their connective tissue surrounding them. These are Hassel's corpuscle made up of lymphocytes. They're lymphocytes and the aggregations of phagocytosed cells. The remains will be found in the Hassel's corpuscles. So that's histological organization of the thymus. Can we be able to talk about it at our own time? You can see now a diagrammatic drawing of the thymus. We can see the capsule sending in trabeculae. This is now a complete lobule. You can see Hassel's corpuscles, lightly staining medulla, and then a darker staining cortex. These are now the Hassel's corpuscles. Secretions, thymulin, thymosin, and thymopoietin. And then now they can be identified by seeing groups of lymphoid tissues, darkly staining cortex, and lightly staining medulla, and Hassel's corpuscles in the medulla. So these are the Hassel's corpuscles, a characteristic present in the thymus. Let's talk about the spleen. Remember, the spleen is the largest discrete organ, largest lymphoid organ. This image is just showing us the parenchymal bit of the spleen. Will come into that in a few minutes. But before then, intrauterine life, it's a site of hematopoiesis. Postnatally now, that's the function of blood sequestration, blood destruction site. Also a blood reservoir, filtration, hemolysis, and production of lymphocytes. So it plays an important function. Intrauterine shows a site of hematopoiesis, site of blood production. Postnatally, it will do the function of blood destruction, blood reservoir, filtration, hemolysis, and production of lymphocytes. What are the components of the spleen? We have the stroma, which will be similar thing as the thymus. But now for this, you'll have a capsule, of course, and then it will send in trabeculae. At this point, the trabeculae will be incomplete. As for example, you can see here is a trabeculae. This trabeculae is incomplete. So it's not continuous at this point. So this trabeculae is incomplete. It sends in, it will divide, the stromal component will divide the spleen into various incomplete lobes and lobules. What about the parenchyma now? Remember for the thymus, we had the cortex and the medulla. In the parenchyma, we'll have the white pulp and the red. So the white pulp will contain all the aggregates of lymphoid tissues. And that is what you can be able to see, the whole of this. The whole of this is actually a white matter, a white pulp. It's the white pulp. For the white pulp, you can be able to see it contain aggregations of lymphocytes here. Those are darkly staining. That is a mantle zone. Then at the center, you have a germinal center. And that germinal center will comprise a structure there, the central artery. The central artery will be surrounded by a darkly staining cells of lymphocytes. So these cells surrounding it, the lymphocytes, making it be called periarterial lymphatic sheaths or the poles. So white matter, aggregations of lymphocytes on the mantle zone. At the center, you'll have a germinal center. At the center of the germinal center, you'll have periarterial lymphatic sheaths formed by a central artery. And then this central artery is surrounded by now these lymphocytes. Apart from now the white matter, the white pulp, so this white pulp is somehow a replica of the cortex in the thymus. But now we also have now a red pulp, which is a replica of the medulla. So this red pulp will contain two things. It will contain blood vessels, yes, that is splenic sinusoids and splenic cords of Birkhoff. So these splenic sinusoids are just spaces. They're just spaces. And splenic cords are just elongations or these sinusoidal cells endothelium which elongate to become the stave cells. So they're actually sinusoidal stave cells are modified endothelium cells. So in addition to that, we're not talking about the red pulp. We need to talk about the splenic microcirculation, the spleen microcirculation. So from the splenic artery, you'll have divided into various branches, splenic arteries, to give you central arteries. So central arteries are now and they are inside the parenchymal. So this central artery will also divide to give you penicillary arteries. The penicillary arteries will divide into sheathed capillaries. These capillaries will become sinusoidal capillaries, that is now red pulp sinuses, which will give you now, in these red pulp sinuses, you can have open circulation or closed circulation. So in open circulation, the blood leaves using these sinusoidal capillaries into the parenchymal aspect, does its work of hemolysis, filtration, blood destruction, production, then takes up the lymphocytes. That is open circulation. In closed circulation, the blood doesn't leave these red pulp sinuses. Then blood from now closed circulation and open circulation leaves using the splenic venules, then splenic veins, then it goes until it drains into the portal vein. So that's splenic microcirculation. This is a brief summary of what we're supposed to talk about. You can see we have a white matter here, a white pulp. This is the white pulp. And finally, it's the white pulp. So this white pulp will contain a mantle zone. And then we'll have this white pulp also containing the germinal center. The germinal center will contain the central artery with periarterial lymphatic sheaths. And then at this point, you can see these are incomplete trabeculae. These are incomplete trabeculae. Trabeculum, they are incomplete. This red pulp now containing the red sinus, containing the red pulp, containing the splenic cords and the splenic sinuses. So kindly Take note of that. And these splenic cords will contain the stem cells. A diagrammatic drawing showing us the lymphatic system of the spleen now. You can see this is a capsule sending in incomplete trabeculi. And this trabecula now at this point, it won't be formed by epithelial reticular cells, it will be formed by elastic fibers. You can see we have this white pulp. The white pulp has the mammalosome and the germinal center. And the germinal center now we have the central arteriole with the arterial lymphatic sheath surrounding it, the lymphocytes surrounding it. And then this is the red pulp now, comprising of the splenic cords and the splenic sinuses. So it will comprise, remember the thymus had a thin capsule, this one will have a thick capsule. Okay, so that's the histological organization of the spleen. If you have any question or any clarification, feel free to ask a question in the comment section. What about now this lymph node? So the lymph node, that's the histological organization of it in terms of it has two parts. It has a medulla and a cortex. For the medulla, similar case like the spleen, it will have now the cords and the sinuses. For it will be known as medullary cords, medullary sinuses. For these medullary cords, they'll just be branching and anastomosing branches of typical lymphoid tissues. So these are actually our lymphoid cords, medullary cords, the lymphoid cords. And then the sinuses are just spaces to allow lymphatic fluid to flow into the inferior end. Then this medulla will contain B lymphocytes, few plasma cells, and macrophages. Similar case, the parenchymal aspect of the lymph nodes contains lymphocytes, a few of T lymphocytes, plasma cells, and macrophages. So they're just immune cells. So remember, you see the lymph node has the medulla and also has the cortex. What about the cortex? So for the cortex, it's the outer portion that is darkly staining. Remember, the medulla is lightly staining. So cortex, outer portion that is darkly staining, it will comprise of subcapsular sinus, you'll see it, a lymphatic nodule, darker circular masses, and T lymphocytes. But before then, let's talk about the stromal part of the lymph node. So remember, first of all, the lymph nodes, they are kidney shaped, they are bean shaped. So they look like it's a bean. So they'll have a hilum, they'll have a concave part, they'll have a convex part. So, but before then, this connective tissue stroma has a capsule which is thin, sensing trabeculi. And then this trabeculi will divide the thymus into the parenchymal thymus into various parts, into various lobes. So between the capsule and the parenchyma, just below the capsule, you have a subcapsular space, which you'll see. Then the parenchyma now, which will be divided into various lobules and lobes by the trabeculi. It will have a darkly staining part, subcapsular sinus, lymphatic nodules, and the T lymphocytes. For the medulla, which is lightly staining, it will comprise of medullary sinuses and the medullary cords. It will contain now reticular fibers, support macrophages, plasma cells, and B lymphocytes. So this is a diagrammatic image showing us the lymph node. So you can see it's bean shaped. You can see it's bean shaped here, or looks like a kidney. But now, of course, these are smaller than the kidney. So this is the hilum with different vessel. This capsule, stromal component, the capsule which is thin. To have on the now the convex side, you'll have afferent vessel getting in. Below the capsule, you'll have the subcapsular sinus. Subcapsular sinus below the capsule. This will be the capsule, so below is subcapsular sinus. Then you can remember this is a capsule sends in trabecula, which will divide the lobe, the parenchymal aspect into various lobes and lobules. But on the sides, you'll have trabecular sinuses. So when now the afferent vessels bring in the lymphatic fluid, into the subcapsular sinus. The subcapsular sinus drains it into trabecular sinus. And then from the trabecular sinus, we talked about the medulla containing the medullary sinuses. So from trabecular sinus to the medullary sinus, from the medullary sinus, not the medullary sinuses, into the efferent vessels. So through this process, now that's where you have filtration happening and the removal of pathogens. The fungal infections, the bacterial infections, and the viral infections will happen there. So let's not talk about now, that's the flow of lymph. Let's now talk about the parenchymal aspect. We have the cortex, which has aggregations of lymphatic nodules. The lymphatic nodules, you can see them here. So just in between the lymphatic nodes, we'll have the paracortex. And this paracortex is thymic dependent because it has a lot of T lymphocytes. So remember the T lymphocytes are coming from the thymus. So its paracortex is the paracortex. The paracortex is here. So it's thymic dependent because it will contain T lymphocytes are coming from the thymus. So you have a deficiency in the thymus. That means you have the paracortex having less T lymphocytes. So that's the histological organization of a lymph node. You can be able to see now the difference between a lymph node and a spleen. So for this, we can go through it at our own time. Feel free to take a screenshot. Feel free to ask any question that you have. So as we try to wind up, we need to talk about now these aggregations of lymphatic tissues in the mucosa, so known as mucosal associated lymphatic tissues, abbreviated as MALT. Mucosal associated lymphatic tissues. So what about these mucosal associated lymphatic tissues? They'll be found in the tracts. So gastrointestinal tract, respiratory tract, urogenital tract. That's where you'll find these mucosal associated lymphatic tissues. Remember the three parts of the mucosa: the lining epithelium, the lamina propria, and the muscularis interna. So in some systems, you'll have just the lamina propria and the lining epithelium. In the GI, you'll have the three: the lining epithelium, the lamina propria, and the muscularis interna. So where you find out these lymphoid tissues, you'll find them in the lamina propria of the mucosa. That's the reason why they are mucosal associated, because they are found in the mucosa. They have mainly T lymphocytes, but not only will they have T lymphocytes, they'll contain immune cells, majorly, so they'll also contain B lymphocyte cells, plasma cells, a few macrophages, um, apart from the GI, where now specifically on the stomach, which will not comprise of these mucosal associated lymphatic tissues. So remember, they are found in the GIT, in the respiratory system, and the urogenital tract. So you can maybe narrow down and call, for example, now we have the bronchus of the respiratory system. So you can call them bronchial associated lymphatic tissues. In the GIT, you can call them gastrointestinal associated lymphatic tissues. So they have specific names in some regions. Some of them in other regions, you just generalize them, but in some, you have to be specific. So that's it regarding the mucosal associated lymphatic tissues. To be specific on this, you can see here, this is a lining epithelium. So this is a column, this is a simple column epithelium with a lot of complex cells there. This is a lamina propria. This is now the lining epithelium, this is a mucosa. The muscularis interna is now internal aspect. So this marks, we found the lamina propria of the colon. Similar cases, esophagus, you can see the lining epithelium is stratified squamous and plasmic epithelium. That's the marks. Below it is a lamina propria now of the having the marks, and then below it you have now the muscularis interna. To be specific now, you have to know these Peyer's patches. They are found in the terminal ileum. The ileum itself will contain the Peyer's patches. Then the tonsils will contain now lymphoid tissues, lymphoid nodules, and remember, MALT consists of small accumulations of lymphoid cells or to a few lymph follicles, as we'll see. And that takes us now to this slide. So take a minute, or maybe you can pause the video and maybe tell us which structure is this. So this, I hope you've been able to guess. So this is the palatine tonsil. You can see the epithelium is stratified squamous and keratinized epithelium. Then you can be able to appreciate below it you have the lamina propria. And beneath it, this lamina propria contains lymphatic nodules, lymph nodes. And then you have this crypt, tonsillar crypts, telling us this is palatine tonsil. The diagrammatic drawing of the palatine tonsil can be able to see this stratified squamous and keratinized epithelium, the lining epithelium of the oral cavity. Beneath it, you'll have the lamina propria, and this lamina propria will contain lymphatic nodules, a few of them, a few blood vessels, and now these are now the mucus glands that you can see there. Okay, so in the next few slides, feel free to pause the video, answer, but I'm going to give the answer. So the first thing, feel free to pause the video and guess, or maybe try to think what the structure is. So for this is a palatine tonsil. You can see lining epithelium, stratified squamous, and keratinized epithelium. Then that's A. For the B is a tonsillar crypt. For E, that's the lamina propria. For D, you can see that's now the lymphatic nodule. D is the lymphatic nodule with C, the germinal center now. And then you have F as the connective tissue surrounding it. And then you have the mucus glands there. Okay, for our next video, for our next image, you can be able to appreciate this is a vermiform appendix. The goal is for us to appreciate this is the lymphatic nodules in the vermiform appendix. This is the vermiform appendix with the lymphatic nodule. So to be specific, at this point, you can appreciate the mucosa, simple columnar epithelium, the lamina propria with the lymphatic nodules. Then below it, we'll have a very small in muscularis interna. Then after that, we have the submucosa. From the submucosa, now you have the muscular layer comprising of inner circular, outer longitudinal. And then you'll have now the adventitia, the serosa surrounding it. So those are the layers of the vermiform appendix. But to be specific, I wanted us to appreciate these lymphatic nodules on the vermiform appendix. This is actually our spleen. You can see this is our white part containing the mondosome and the germinal center. And then on this germinal center, you can be able to appreciate at this point, we have the periarterial lymphatic sheaths, white pulp, germinal center, periarterial lymphatic sheath is here. Is a white pulp, germinal center, periarterial lymphatic sheath is here. Periarterial lymphatic sheath is here. So then you'll have now this red pulp containing the splenic cords and the splenic sinuses. For this, these are lymph nodes. Aside of the lymph node, you can see the capsule. There's a capsule. This is now the cortex. This is the medulla. And then now you can appreciate this now the hilum, the region around the hilum. So remember what we talked about this. What about this? This is the thymus. Yes, you made the correct answer. You can see it's a trabecula dividing into various complete globules with now outer medulla, outer cortex, which is darkly staining, and another part which is lightly staining. And here you can see a scopus. For this, feel free to unmute. I'd like us to start with this. So, what is this? I hope you made the correct answer. This is a lymph node. This is a lymph node. You can see we have the cortex there, and there's a medulla. You can see the aggregations of the lymphatic nodules. So this is a paracortex. This is a subcapsular sinus. And then in this capsule, you can see our blood vessels. So sending in trabecular sinuses, here we have the medullary sinuses, likely around this place. You have now the efferent vessels. So these are lymph nodes. So from this image, I would like us to take us to this image. So from here to here, So this is now the efferent vessel of the lymphatic fluid. So this there, the medulla. So it's a higher magnification of the same. And then it's showing us the red bulb. So here you can see the sinus and the cord surrounding it. Sinuses surrounded by cords. So these three images showing us the lymph node. Low magnification, higher magnification. For this is now our spleen. This is our spleen showing us the parenchymal aspect, incomplete trabeculi. These are the white pulp, white pulp, white pulp, and then you can see the red one here. Here's our red one, and that's a slide on the spleen. Comes a lot in exams. What about this? I hope you made the correct answer. That's the palatine tonsil. We had gone through it, so I won't belabor much on it. What about this? That's the thymus, and in this thymus you can see complete lobes, globules. That's the cortex, the medulla, and you can see the hasos corpuscles there. Another hasos corpuscle is here. Okay, what about this? I hope you made the correct answer. This is a spleen. Yes, this is a spleen. You heard me correctly. These are now incomplete trabeculi. At this point now, for the spleen, remember it won't be that clear, but this now the regions around the red part, then the white part will be around here. But the correct image that I would like us to get it is this. This comes a lot in exams. So for this, there's a spleen. What about this? For this, it's actually higher magnification of the same. I would like us to appreciate that all of it is darkly staining. Then you have this is our germinal center. And then at around here, now we have the peri-arterial lymphatic sheets. So this also on the spleen. So this actually the whole of this is showing us the white pulp gives a division around so the mantle so around here, then the germinal center around here, and then this is peri-arterial lymphatic sheet, the central artery being surrounded by peri-arterial lymphatic sheets. So for this, I hope you're able to talk about this in the thymus. Yes, this is the thymus. This is also the thymus. You can see complete lobes, complete globules. Then that's the hasos corpuscles. That's a darkly staining medulla, lightly staining, lightly staining cortex, lightly staining medulla, with the hasos corpuscle being found here. So similarly, I wanted us to appreciate a couple of these medulla now on thymus. This hasos corpuscle in the thymus. And now these two images are showing us the spleen. I hope you're able to identify this as the white pulp with the central artery. So the whole of this is actually the white pulp. This is a red pulp here. White pulp. This is now the peripheral lymphatic sheets. Incomplete trabecular. Yes, so that is it. This was a slide on the spleen. The spleen. And then I hope you can be able to identify this. It's actually being shaped. This is the hilum with efferent vessel. Then at this point, you can see the capsule. This is actually the cortex, the medulla. So which organ is this? I hope you're able to answer that this is the lymph node. Yeah, and then you can see now how you have now the medullary sinuses coming in, draining into the efferent vessel. So these around the concave parts, then the convex with the afferent vessel, the concave with the efferent vessels. So these are a few sample essay questions which you can try at our own time. What are the two defense mechanisms mediated by the lymphocytes? So talk about humoral and cell-mediated, two sequences of the sequence of flow in a lymph node. We discussed that. What is the thymus dependence on in a lymph node, the paracortex? Why? What are some of the antigen presenting cells? Talk about the dendritic cells, the macrophage. What are those antigen presenting cells? The neutrophils and the rest. What are the two components of the red pap of the spleen? So we talk about the sinuses and the cords. How do you identify the white pap for the spleen from the lymphoid follicles of a lymph node? So how do you do that? I presume we covered that. What is the thymus dependence on the spleen and why? Describe the two types of circulation in the spleen, that is closed and open circulation. What are stem cells of the spleen? What is their functions? Name the functions of the various epithelial reticular cells of the thymus. Remember, there are 6 cell types, 6 epithelial reticular cells. Type 1 and type 3, they form part in the blood thymus part. What about the rest of the 4 cells? So kindly go through them and list them. And then what is a hostoscopus, call its function. And I guess with that, the last question will be name the structure that form the blood thymus barrier. So I guess with that, just to say thank you, kindly feel free to like, share, subscribe with telling about your friends. I'll see you in the next video, which will be now on the histology of the GIT. So I guess with that, have a wonderful time ahead. Thank you.